Hi all, hope you all are doing good. So today we are going to learn about pharmacology. So what is pharmacology? Why even learn about pharmacology? Have you ever imagined the medicines which our doctor prescribes us for curing our diseases, from where they are getting derived? How those medicines are getting the ingredients derived? Have you ever wondered about that? We just go to the medical shop and the pharmacist gives us the medicines. We take those medicines, we come to our house, we swallow those tablets. But have you ever imagined or even wondered how these medicines are being prepared? How they exactly know by using those medicines our symptoms will reduce or vanish. So those ingredients, how they are derived, how that drug once it enters our body, the medicine when we swallow it, how it is solving our issues, our symptoms or our disease, how that is happening. Then how our body is handling those medicines once they are inside our body because it's a foreign substance. Now your when it enters our body, it might affect us if it continues to stay inside our body. So how body is no sending those observed medicines into different parts of our body. So in today's session we'll be seeing all that. By the end of the session you will know how the medicine which our doctor is prescribing is getting derived. So before even starting to know understand what pharmacology is, let us go through some of the terminologies which will make you to understand the definition of pharmacology better. So the first one which comes is chemical substance. So what is this chemical substance? So chemical substance is nothing but it is any matter. Maybe it's solid, liquid, gas, anything, any matter. So we all know matter is something which has mass and it occupies space. So any matter which has a specific or constant chemical composition and distinct properties. For example, what they mean by constant chemical composition means if water is there, the formula is H2O. So that will never change. It is constant. Two hydrogen atoms and one oxygen atom. So that H2O molecules will never change. Similarly, for salt, we have NaCl, sodium chloride. So, sodium and chloride will never change. That combination will always produce salt. So, so chemical substance is any matter which has constant chemical composition. Similarly, our lead, it is a metal, silver like metal, lead. So all these chemical compositions will never change. So these are all chemical substances. All the things around us, common things, all are chemical substances, like even plastic, your oxygen. Those are also constitute the chemical substance. Next, it is about drug. What is a drug? So, drug is any specific type of raw active chemical substance. Now we have seen what a chemical substance is. So, drug is any chemical substance which has the power to alter a biological function. Okay, If it does not have the power to alter a biological function, then it will not be called a drug. Okay. For example, if any chemical substance has the power to lower our blood pressure, okay, then that will be called a drug because it is changing our biological function. It is affecting our biological function inside our body. So, drugs, example, they can be no helpful, harmful, or neutral. So, example, if you see alcohol, nicotine, caffeine, and some drugs like captopril drug from snake venom or synthetic drugs which are manufactured in the lab, invented in the lab, all these constitute drug because they have the capability to change the biological function inside the human body. Okay, so that is a drug. Next is your medicine. Now we have seen chemical substance, drug, and now comes the medicine. Medicine is any drug. Okay, we saw what a drug is. So, any drug that has been specifically prepared, dosed, and has been proven safe and legally approved to cure a disease or prevent a disease is called a medicine. So, these are all raw drugs, raw materials. Now, these drugs, if they have been proved, they are safe for being administered to a living being without any or with minimal side effects, and it has been approved by government authorities, as well as it has been proved that this drug is safe. So, after going through all those steps, the above drug becomes a medicine. For example, for medicine is whatever medicines we are using, your paracetamol, your metamorphin. Metamorphin is used for your sugar patients, where your diabetic patients are using these tablets. Ibuprofen capsules, these are used for reducing inflammation. So, all these certified drugs, we can say, can become medicines. So, as we have seen all this, now we can conclude saying that all the drugs are chemical substances, but not all chemical substances are drugs because all chemical substances do not have the ability to modify the biological function. Only if it modifies the biological function, it is called a drug. Okay. Now, similarly, all medicines are drugs, but not all drugs are medicines. See, whatever medicine is derived, it is derived from drugs, but all drugs cannot turn into medicine. Some drugs can be poisonous, so they cannot use as medicines. So, this is these three terminologies are very important as this will help us in understanding the definition of pharmacology. Okay, so now if you see, now we'll try to see and understand what pharmacology actually means. Okay, so if you see here, pharmacology means it is deriving from Greek words. So they are just splitting that word into pharmakon and logia. So pharmakon plus logia gives pharmacology. That is how that word was derived. And pharmakon means drug or poison. And logia means the study of. So, if you take the literal meaning of pharmacology, it comes out to be it is the study of drugs and poison. So, pharmacology is the study of drugs and poison. This is the definition we get by looking at the Greek words. So, now we will see the actual definition of pharmacology. So, pharmacology is the scientific study of how chemical substances, we saw what are chemical substances, and drugs interact with living organisms. So we have chemical substances, it can be anything, like we are interested in knowing about lead, cyanide, carbon monoxide, all these are chemicals. And drugs like caffeine, paracetamol, how they affect our biological functions, their interaction, what they do to the living organisms. It can be birds, animals, human beings, anything. How these substances interact with the living organisms is what constitutes pharmacology. So, under this huge subject, pharmacology, these things are studied. So, if you see here, we have included even the chemical substances, not only drugs, because we are also interested in knowing what happens when any poison enters our body. Like carbon monoxide has poisoning effect, lead has poisoning effect, cyanide has poisoning effect. So by studying about these chemical substances and drugs, we can understand how they affect our human body. So our poisons, all these things, are toxicology is one more branch which is there under pharmacology, where a separate branch is studied to know about the effect of poisons in the human body, the toxicology, all toxins. So when we say chemical substances, it does not only mean the poisons, it also means the naturally occurring chemicals, which molecules which are present in our human body. So we already have naturally occurring chemicals. Only if we understand that chemicals, we can prepare our drugs so that they can change our biological functions. So pharmacology is the scientific study of how chemical substances and drugs interact with living organisms, and chemical substances include both the chemicals which are present inside our human body, naturally occurring chemicals, as well as the chemicals outside the human body, like your carbon monoxide, lead, and cyanide. So, this is the definition of pharmacology. Now, we will see there's a lot of confusion which happens when we say who is a pharmacologist, who is a pharmacist, and we also have the pharma companies. So, we'll try to understand the difference. So, what each person does will become clear for you. So, basically, a pharmacologist, which we are studying now, the subject pharmacology, he's a pharmacologist. So, what he does is he's a scientist who works in a laboratory, who studies the chemicals, drug interactions with the living organisms, and he discovers and invents drugs, as well as does the safety check of the drugs at the laboratory level. So, safety check at the laboratory level is nothing but he does the safety checks on animals like guinea pigs, rats. So, they experiment those drugs on those animals and confirm that drug is safe or not. So, pharmacologist is that person. Now, pharmaceutical companies, these companies, once the pharmacologist has discovered the drug or he has invented the drug, they take the new drug for human trials. He has proven safe on animals, it is taken for trial on humans. which is sponsored by this pharmaceutical company. So many people are tested, volunteers, with this new drug, and some people with the disease are tested with the new drug. So once this drug is proven safe, they do the bulk production of medicines. So how they do bulk production of medicines? The drug cannot be sent into the body as it is. It has to be binded with some binders, some safe binders. These binders have also been are tested and proven safe. So, like some glue kind of structure or glue kind of material or like a fiber, so they mix with these medicines or the drugs which now the pharmacologist has discovered. They mix with that and produce the final product which we get in the uh, in bottle form or strip form. Those are the medicines. Or we also have like an ointment, we have inhalers. So all those medicines bulk manufacturing is done by the pharmaceutical companies. Okay, next, from here the medicines are sent to the pharmacist. So pharmacist acquires the medicines and he is the one who dispenses the medicine. So pharmacist, see, is completely different from pharmacologist. He invents medicine and he dispenses medicines. So dispenses medicines in the sense he gets the prescription, the doctors, he takes the doctor's prescription, he sees the prescription and sees the medicines which have to be given. No, sometimes you would have seen if the medicine is not there, he gets another, he gives us another brand medicine. So he basically dispenses medicine. So he's a pharmacist. So hope you have understood the difference between these three. So next we'll see, now we have seen the drug. So which is the final product which pharmacologist invents and gives it to pharmaceutical industries for manufacture of medicine. So how these drugs or the raw materials for these drugs are obtained, from where we get these ingredients, no, or where we get these drugs from. So drugs are the raw materials. So from where we are getting those raw materials, we are going to see that now. So if we see the raw materials, the drugs are derived from natural sources, synthetic sources, semi-synthetic sources, and biosynthetic sources. So now we will see what are the different sources and how they are getting those drugs. So natural sources. If we see natural sources, how they get the drugs from these natural sources? Like plant sources are one of the natural sources we get from the bark of the tree or from the roots of the plant, from the seeds. All these are got from these plants. For example, quinine is a drug which is used for malarial treatment and that is got from the cinchona bark. So cinchona tree's bark we get this drug called quinine which is used for malarial treatment. And another way we get these drugs are from microbiological sources like fungi and bacteria. We get those sources chemicals, the drugs. So penicillin is there, right? So penicillin is got from this Penicillium notatum fungus. So we get from that fungus we get this medicine penicillin. So that is a chemical which is produced by bacteria and fungi. And we get some more natural sources like from animals also we get some drugs like for thyroid medicine we are getting that from the sheep. From here it is source, and the antidote for snake venoms are got from the snake venom. So peptides from the snake venom we get the antidote prepared from the snake venom itself. So we get that drug from there. Next is mineral sources. Mineral sources is we are having many elements which are found on the earth. So iron is mined and refined to treat anemia. So iron tablets which we are given when we have anemia or low blood, that is derived from iron which is mined from our earth. So these are the natural sources from which our drugs are derived. Next are the synthetic sources. So synthetic sources are something which are completely man-made. They are not found in the nature anywhere. So they have to be prepared from scratch in the laboratory. For example, your aspirin tablet is there, your paracetamol tablet, your BP tablets. All these are manufactured from the scratch in the laboratory. So, next is semi synthetic sources. So, semi synthetic sources is nothing but half synthetic, half natural. So, what happens here? A natural source is modified to produce the new drug. So the penicillin, which is derived from fungi, which we saw, that is modified in lab to produce amoxicillin. So amoxicillin is produced from penicillin. So we get that penicillin, go to lab, modify it, and we get amoxicillin. That's why it is semi-synthetic sources, lab altered natural sources. Next we have biosynthetic. So, biosynthetic is we are doing genetic engineering, that is biotechnology. What we do here, the drugs are prepared by cloning of human DNA in certain bacteria or yeast cells. Example, the insulin which we use in diabetics, that is derived like this. So, what they do, here we will see that the genetic engineering process, how they derive it, the drugs. So now what they do, they take our human DNA and they will search for the gene which they require. For example, I want to know manufacture insulin, so I will check in the DNA for the insulin gene and that one will be copied. Okay, and kept, a copy of that is kept. Next, a bacteria is there. In that we have plasmid DNA, they have their own DNA. The another DNA is plasmid DNA, which are no circular in structure. That DNA, it is cut at particular places with restriction enzymes, so that this gene can be inserted in that place. So they insert that gene and now it becomes recombinant plasmid, the new modified plasmid. This is inserted into the bacteria. So after this, what happens? The bacteria replicates and produces the insulin hormones which is required. So it generates insulin and that insulin is as extracted and used in our medicines. So this is called genetic engineering. This is how the drugs are no derived. So this is how we derive the insulin drug. So this is also one of the method by which our drugs are derived. So next, if you see, now we are going to see the two main branches of pharmacology. Till now we saw the sources of drugs. Now we will see the two main branches of pharmacology. Now the study on the drugs is done. So there are two main branches called pharmacokinetics and pharmacodynamics. So pharmacokinetics is nothing but what the body is doing to the drug. So body is there, how it is interacting with our drug. It can be any drug, for example, paracetamol. Next is pharmacodynamics. What this drug, once it enters our body, is doing to our body. That is pharmacodynamics. So, drug is there, paracetamol you are swallowing. So, what it does to our body. So, that is pharmacodynamics. So, now we are going to see each of these branches in detail. So, let us go to first one, pharmacokinetics, and try to understand that. So, what is pharmacokinetics? Pharmacokinetics is the study of what the body does to the drug. Okay, so here what the body is doing to the drug is going to be studied here. Okay, so here is a typo, so let me correct it. So here the drug is going through four processes in the body. So basically what happens when you are not swallowing the drug, it goes into our body. So when it enters our body, it goes through four processes inside the body. Four different stages it goes through, like absorption, distribution, metabolism, and excretion. So that is simply summarized as ADME. That entire pharmacokinetics. So that is what we are going to see now. Now let me zoom in. Okay. Yeah. So now this is the drug. It is entering our body. Okay. Then we have absorption, metabolism, distribution, and excretion. So whenever a drug enters our body, it goes through all these four steps. So we will see each of these step in detail. So first we'll see the absorption. So in absorption process, if you see, I'm having the drug, okay, it enters our body, esophagus, it enters the stomach, it gets mixed with the stomach acid, then it enters the small intestine, then the large intestine. So the drug gets absorbed in these intestines. So it gets absorbed via this route. Now what happens? The drug has been absorbed and it enters the bloodstream, that is the hepatic nerve, hepatic vein, which is the vein which is connecting the liver. So now liver is there, right? The liver acts as a checkpoint. It's the security guard. What the security guard does? The security guard checks for any foreign particles inside the blood before it is sent to the heart. Okay, so you have understood till the absorption process. Stomach, food you are eating, stomach, it reaches small intestine, large intestine, it is observed. Absorption is done. It enters the hepatic vein, which is connecting the liver. So any blood or anything which is observed in small intestine and large intestines is not directly sent to the blood, is not directly sent to the heart. It always has a checkpoint, that is our liver. So what our liver does, it checks for any foreign particles. Like our drug is a foreign particle, so it tries to destroy the drug as much as possible. So after going through that metabolic activity, metabolism, that is called the first pass metabolism, because that is the first step when the drug goes through the metabolic cycle. So after it no destroys as much as drugs from the liver. Now the liver sends the blood to the heart. So now heart gets the blood. So by the time it reaches the heart, the bloodstream, um, your drug, it is destroyed by 70 percentage. Already only 70 percentage of drug is left for it to enter the bloodstream. Now from the bloodstream, from the heart, what happens? The heart pumps the blood to all parts of the body. So now comes the distribution phase, where from the heart the blood is pumped to all parts of our body, and it enters all our small tissues, organs, everywhere the blood flows. So what happens now? The drug, 70% drug or whatever remaining drug is there, it enters all parts of our body, and your distribution phase is completed. So it enters the different parts of the body and reaches the target site. That happens during your distribution phase. Okay. Now it has reached the target site. It has done its duty, the drug. Now what happens? Now what we have to do? We have to remove the drug out of our body. Okay. Now drug has to be removed. So what it does again, from all these organs and tissues, the whatever remaining drug is there, it gets sent back into the bloodstream. And the bloodstream again, goes through this liver, and after it enters the bloodstream, it goes through the liver, and it liver does the metabolism. What type of metabolism? Now it does the metabolism to remove these foreign particle, which is the drug, from our body. So in case your drug is fat or lipid soluble drug, it changes it to water soluble so that it can be eliminated from the kidneys. So kidneys no cannot filter the lipid based drugs, so it changes that, modifies that into water based drugs, and it enters the kidneys and it is excreted out. That is one way of excretion, that is your renal excretion. One more thing is the liver, whichever is left over, that drugs, what it does, it pushes it to the bile ducts, which are present in the gallbladder, and that pushes it to small intestine, and it goes out via the fecal route. So even the liver as well as the kidney help in the excretion of the drug. So this is the pharmacokinetics process, the entire process which happens inside the human body with the drug. This is what the body is doing to the drug. So we will see, here everything in detail. So we saw absorption, the drug enters the bloodstream from the administration site. Bioavailability, this is, I was telling, the drug, by the time it reaches the bloodstream, most of the molecules are lost because the liver does the metabolism. So by the time it reaches the bloodstream, only 70% of drug is left. So that is called bioavailability, how much amount of drug is left. So the administration site can be your mouth through tablet form, or it can be IV injection, where directly in the blood vessels the injection is given. So if the injection is given right, IV injections, that will directly be and that will directly enter your bloodstream, and your bioavailability of the drug will be 100%. So none of the drug will be lost. That's why no, mostly when the injections are given, the dosage will be less compared to when we are given through mouth. The tablets dosage, if it's 500 mg, your injection dosage will be only 10 mg or 100 mg, because that is the bioavailability is high for that. It's 100, almost 100%. That's why for injections the dosage is always low. So we saw the distribution phase, where see distribution phase is the reversible transfer of the drug between the blood plasma and the body's tissues or intestinal fluids. So in distribution phase, what happens is we saw the blood is transmitted, no? Drug is transmitted to the blood throughout the body. So what carries that drug? It's the blood plasma which carries that drug throughout the body. And if you see here, there is a word called reversible. So reversible simply means it can transmit from the blood vessels to the body tissues, from body tissues to the blood vessels. So it can flow in both ways. If this was not reversible, what will happen? Everything will get accumulated in our blood and it will lead to toxicity or poisoning of the blood. So that's why distribution is always reversible. So that's why I say the both side arrow is there, blood to tissues, tissues to blood, double side arrow. So how that happens? Initially, the blood has high dosage of the drug, so it moves into the tissues. As the organ clears the blood, so once it enters the kidneys, liver, no, it is no clearing or the organs clear the blood in the sense they no longer require the drugs. So here the symptoms or the disease is getting cured, so the concentration of the medicine is reduced and the concentration of the blood becomes low, so it again moves back from the organs to the blood, because blood is giving the passing the drug into the organs, so concentration of the blood will reduce. Then once that is happening, it will push back the um, drug from the organs to the blood. Next, if you see the metabolism, which we were discussing, happens in the liver, and the first pass metabolism is what we discussed, which happens before our distribution part, where I told it happens before distribution and after absorption. We saw the liver acts as a security guard there before entering the heart for the entire distribution of blood. That's called the first pass metabolism. Next is your systemic metabolism, which is the usual metabolism which happens after the distribution phase, so that the chemicals are removed from our body, like the lipid soluble drugs are modified into water soluble drugs. And finally comes the excretion part, where the elimination is irreversible. It will not come back once it is no sent outside. It will not come back. So that's why it's called irreversible removal of the active drug from the body via the kidneys and liver. So that is the excretion process. Now, for the oral pill, we have seen absorption, metabolism, first pass liver, then distribution happens, then metabolism, and then excretion. And the oral pill is removed from our body. So that entire process is given here in short. See, mouth, enters stomach, intestine, portal vein, the vein blood vessel which we are discussing, which connects the liver. From there, liver does security check, the first pass metabolism. It sends to the heart, then it enters the bloodstream and organs and tissues. After everything is over, it is pushed back into the bloodstream. Then it enters the liver for the breakdown of the remaining drug present in the bloodstream, and finally it is excreted via the kidneys and the liver. So this is the entire process of the pharmacokinetics. So hope you have understood till here. So see how our body is no handling these drugs inside our body. So if in case you are being given an IV injection, so IV injection, intravenous injection, here you see absorption is immediate. Directly it enters the bloodstream. There is no absorption happening separately. It directly enters the bloodstream and it's distributed to the body. Then metabolism happens and excretion. Here we don't have that first pass liver, no security checks are here. Directly it enters your bloodstream. That happens with the IV injection. So hope you have understood what our body is doing with the drug. Now we will see what um, no the drug which we were seeing is doing to our body. That is pharmacodynamics. So what is pharmacodynamics? So pharmacodynamics is what the drug does to the body. In short, it explains how the drug cures a disease or relieves symptoms once it reaches the target tissue after the distribution phase in pharmacokinetics. In pharmacokinetics, we saw first absorption happens, then distribution, metabolism, and then excretion. So now where this pharmacodynamics comes in that phase, it comes after absorption, then distribution. So after the drug gets distributed to the organs and tissues and it reaches the target tissue, there comes the pharmacodynamics. So here is where the pharmacodynamics plays the role. So we are going to study about what the drug is doing to the body once it reaches the target location. Okay. So if you see here, so here the drug is arriving at the target tissue and then it is binding with the receptor, the cell response, and then we get the symptom relief or cure. So this is the entire process shortened, but we will be discussing that what is happening each and every step we are going to discuss. So whenever no drug has to do something inside our body, it always has to have something to bind to or something it can attach to. Okay, that is your receptor binding. So target tissue arrival, whichever tissue is having issue, it will arrive there. Then comes the receptor binding. So whenever any chemical enters our body, if it has to act on our body, it has to have some target where it can attach itself so that it can do what it needs to do. So that is your receptor binding. So the core principle of this pharmacodynamics is your drug receptor interactions. How the drug and the receptors interact. We are going to see that now. So as we have discussed just now, drugs work by binding to specific cellular targets called receptors. So there are specific targets which are present on the cells on which your drug is binding. Those are called the receptors which are present on the cell, right? Those are nothing but those are kind of proteins. Okay, there are no proteins which are present on the cell, which these drugs come and attach to those receptors and send the signals what the cell has to do. That is what is going to happen. Imagine no, your receptor is like a lock and your drug is the key. If you give the key, the receptor will open the lock and do its duty. If you are jamming the key, nothing will happen. So drug is the thing which binds to that receptor. So we are going to see the drug receptor interactions. So what happens? The drug molecule comes, whichever drug no is being no manufactured or produced. That drug will combine with the cell receptor. Okay. Now what happens? Now the cell and drug receptor complex is formed. So cell and receptor complex is, they both combine and form that new complex. After that, the cell acts as per what the drug and receptor complex tells it to do. It sends signals or it blocks something, it does something. The drug, once it binds with the receptor. So that is what happens while doing the drug receptor interaction. Now what happens is, there are three types depending on three types of drugs, how they, based on how they interact with the receptors. Okay, so you'll able to understand this. No, I will show you diagrammatically so that you understand it better, what is this drug and cell receptor interactions. So now, based on this interaction between the receptor and the drugs, there are three types of drugs. What are the agonists, antagonists, and partial agonists? Okay, so what happens now? Um, so we have the antagonist, agonist, and partial agonist. So what happens? Now we can see here. Okay, now this is an agonist. So agonist, what happens? This is your drug, and this black color one is your receptor. So this is your cell, your target cell. So just know that receptors are nothing but some protein structures with a particular shape so that they can fit in the naturally occurring chemicals inside the human body. So this agonist is a drug which are prepared mimicking the natural chemical present, okay, inside the human body, okay, and so that they fit exactly inside this receptor. So now agonist is now going and sitting inside the receptor. What happens? It fully activates the cell and asks it to do its duty. So we see it is fully activated. That is an agonist. So antagonist, what it does, it just goes and sits there. It blocks it. This drug just blocks it. It will not allow to do anything. It's like a blocker. So the cell will not be able to do what it has to do because this has blocked antagonist, and the natural chemical cannot sit here because this has already been occupied the receptor. So that is what an antagonist does, is it will block that action. We will see what examples for that. Next is your partial agonist. This will partially uh, no activate the cell to do its duty partially, not fully, partially. Now for example, if you see here, if the morphine drug is there, okay, that is basically given for pain, okay, to turn off the pain signals. So morphine is a drug which goes, it mimics the naturally occurring chemical morphine, and it goes and binds to the receptor. So what happens? And it activates the cell, so it will stop the pain signals. That is what this does here. So we saw the morphine will be doing those things. Next is your antagonist. What it will do, it will go and sit there and it will block it, all right. So beta blockers are there which sit on the heart receptors and they block adrenaline, because of which the heart rate is lowered. So they block. When this adrenaline is released, we know the heartbeat races and it beats fast. So now what is happening? This drug antagonist will go and sit here and block these receptors from no the other chemical molecule coming and sitting here and activating the cell. It will stop the cell function so that it will not generate adrenaline and your heart rate will remain normal. So that is what an antagonist done. And partial agonist is he will release partially. So the drug is designed such that it will not fully activate the cell, it will partially activate the cell. So now we'll see the how no they work, how these different mechanism of action happens. So we already know that the drugs generally interact with existing target structures like proteins are there inside our body, enzymes are there, or receptors are there in our body. So drugs will interact with these existing target structures to bring the biological function change. So we'll see what is their mechanism of action. One we have already seen the receptors on cell membrane. So here what happens, the drugs go and sit on the receptor of the cell membrane. So what are these proteins are purely for communication and nothing else is done by these receptor proteins. So drug goes and it communicates to morphine to turn off the pain signals, and the morphine is the drug here we are using and it will turn off the pain signal. This is one type of mechanism by which the drugs act. The other things with which the drug interacts is the enzymes. So here enzymes are the receptors. So how it does, what are enzymes are catalytic proteins where they speed up any chemical reaction. So this is the drug, it will go and sit in the active region of the enzyme. So now what happens, it has occupied the place, so no other chemical can come and enter the enzyme, so it will block the enzyme's action. For example, aspirin is there. This, if aspirin is sent, it will stop the COX enzymes from to stop the production of inflammatory chemicals. So this will come and sit here. So this enzyme will no longer able will be able to produce the inflammatory chemicals. So inflammation will reduce. So similarly, if you see the another form, what they do is the drug goes and attached in the other place of the enzyme, not in the active but in other place, so that the structure of the enzyme changes such that these enzymes um, or the substrates they are called, these are, for example, lactose is a substrate which enter this enzyme so that they can no modify this, get modified, the substrates get modified and get released into the blood. So now sometimes no drugs are used so that this activity is increased. So now many substrates will come and sit and they will get modified and released into the blood. So this enzyme will generate more output compared to what here it is blocking the output, here it will be generating more output. So like that also the drugs can be used. Next is the ion channels as receptors. So ion channels are nothing but no, we are having ions like sodium ions, potassium ions which enter the cell membrane. So these proteins are controlling the entry of these ions. So by using these drugs, we can open these protein paths so that the ions can enter this cell membrane. So that is how the ion channels act as receptors. So this is how the drug can act. The other one is the non-receptor actions. Now there is no receptor present. So how the drugs are acting? For example, we have the antacid. Just by the chemical properties, they act in the body. We drink that, and the acidity in our stomach is reduced. So they just act as direct no basis by chemical simple chemical reactions. They reduce the symptoms inside the body. So that type of drugs are also there. They are non-receptor actions. That is how the drugs can act. So now we will see what are the important pharmacodynamic concepts. So first one is comes the efficacy. Efficacy is nothing but it is the power of the drug. Power of the drug means maximum relief it can provide irrespective of the amount taken. So if you are taking aspirin, it has low efficacy rate. Means how many ever aspirin tablets you take, your pain will reduce only to a certain extent. So that is what is efficacy. So if morphine tablet you are taking, okay, the morphine tablet has high efficacy. Why we are telling like that? Because it has high pain relief. So if you are taking morphine, the amount of pain relieved will be higher compared to the amount of pain relieved by having the aspirin tablet. So that is the maximum amount of pain it can reduce, not more than that. So that is what determines your efficacy. So with the same dose or multiple doses, it can't reach or cross that maximum which it has. So that is called high efficacy and low efficacy of the drug. Next is potency. Potency is how much no small dosage of the tablet can get you a result. For example, if you are having one small tablet and it is no giving no high relief, and we are having another tablet which is bigger in size and it is giving the same relief, but depending upon the size or the what to say the amount, the dosage. So the tablet with the lower dosage giving uh, the same result, giving a result is called more potent. For example, if you are having one candesartan tablet, 8 mg tablet, if you take that tablet, okay, your blood pressure is controlled. But for the same thing, you have to eat low certain 50 mg tablet. So see, your dosage has increased. This tablet, if you take, you have to take 50 mg. So if you take 50 mg only, your blood pressure is under control. So now, when we see both and compare both, which is more potent? The 8 mg tablet, even with a small dose, it is giving higher result. So that's why it is called more potent and compared to the other tablet. It is called less potent. Next is your therapeutic index. What is this therapeutic index? Therapeutic index is nothing but your safety zone. That is the gap between a helpful dose and a poisonous dose. We know any tablet when no it is taken in higher doses instead of one tablet. If you eat 20 tablets, you will have side effects and also poisoning might happen. So that's why therapeutic index determines the gap between a helpful dose. Okay, one tablet is curing your fever. So what happens if 100 tablets are curing my fever or not? No, even if they're curing, are they turning toxic inside my body? Means if I eat one tablet and by mistake I'm having two tablets, how much level of tablets the body is tolerating? After what level that turns into poison? So the gap in between that is called your therapeutic index. For example, if one pill is helping us and 100 pills, if we have the same tablet, 100 pills of the same tablet, it turns into poison. But you see the gap is huge, high safety zone. So maximum I can have 100 tablets, till that nothing will happen to me. It will not turn into poison. So therapeutic index will be how many tablets will result in poisoning and how much will help us. So this will be give us a therapeutic index of 100. So therapeutic index always should be higher so that the drugs are safe. Okay. So even a small change in pill will create problem if the therapeutic index is low. We'll see the another example here. Now see if here one pill is there, it is helping, two pills will turn into poison. See how much low safety zone is there. Even if by mistake I'm having no second tablet, that's all. It will turn into a poison. So it has therapeutic index of 2. So these tablets we should be very careful. These are very high dosage tablets which will no having a therapeutic index low will result in poisoning, more toxicity compared to these drugs where you till I have 100 tablets I will not have any poisoning occurring in my body. Now we have seen both, right? So depending on that, you have two types of therapeutic index. Wide therapeutic index, they are considered as the safe drugs. For example, because of the high safety zone. See, one tablet I have and I have 100 tablets here. So we are getting high therapeutic index. Example is amoxicillin, the antibiotic amoxicillin. So even if I have no 2 3 tablets, uh, it will not create that much poisoning compared to the low therapeutic index drugs. So this is having high safety zone. It will not create any poisoning in my body. Next is narrow therapeutic index drugs are called dangerous drugs. Why? Because the safety zone is less. Even by mistake, if I'm going to have a second tablet, then it will lead to internal bleeding. Example is foreign blood thinner, which is given no, for preventing clots in the blood. One tablet is fine, but if I have 2 tablets, then it will result in internal bleeding. So that's why these are all dangerous drugs. We have to be very careful with this drugs which have narrow TI. So most of the drugs are considered safe when even high dosages will not affect our body, cause poisoning in our body. So hope you have understood this pharmacodynamics and pharmacokinetics in this pharmacology subject. So that is the reason I think by now you will have understood how these drugs are no got and how they affect our body. So hope you have enjoyed today's session. So if you like my videos, please like, share and subscribe. I will come up with another new topic for my next session. Thank you all.